And without further ado, I would like to hand it over to JJ to tell us a little bit more about Young's Farm.
Yeah, thank you for that introduction. So like Whitney mentioned, we do have a trial going on. It's actually right behind me where we're doing 4 different no-till methods. One is solarization, one is roller crimping, and then one is also like a semi-permanent bed system. And then we have a control group. In our no-till greenhouses that are permanent bed systems, we've gotten pretty good at managing that. So we want to start taking it outside, and instead of converting the entire farm over to a no-till system, we want to do a small kind of 4-acre trial, and then we'll expand from there. The Young's family has been a conventional farm for probably the last 100 years. And in the last 5 years, we've been transitioning the farm over to an organic operation. We did just go for our certification, and we are waiting for approval right now. And within that, we are planning to expand as well to eventually a 60-acre farm, hopefully.
Thank you, JJ. And I would also like to point out that the background, JJ's background currently, is the field that we sampled soils from. What are you growing in that field?
So right behind us, we have a rye, vetch, and clover in the field right now. There's been about 1 ton of compost spread on each field. Also 200 pounds of 600 with a zinc and boron additive that we needed as well. And then when I got here, most of our soils were under 2% organic matter, so they were very infertile. After getting our soil test back, we're ranging into a 4 to 6% across the entire farm now.
Excellent. So now we're going to take a closer look at those soils. And I'm hoping that Monique can join us as well. So here we go. We have the 3 soil samples and a line here. Can everyone see my screen okay? Awesome. Okay, so the first sample is a recently and historically tilled field. Jonathan can perhaps add a little bit more detail here, but he did mention it was one of his most difficult blocks that had perhaps consistently the worst soils. And then the middle sample, which looks distinctly different from the other 2, is from a no-till greenhouse setup that when we sampled it was in the process of growing some leafy greens. Was it arugula?
Yeah, we were in our mixed greenhouse at that time.
Excellent. And the third sample, all the way to the right, was from what we would call the hedgerow or the edge of the field. So it was relatively untouched, uncultivated, and so on and so forth. So this is our first sample, again, a more recently tilled field. And this is at our lowest magnification that you'll see today.
All right, so I'm going to pipe in here about the microscopy we're looking at. So it looks very full. You can see it's full of bacteria. The only thing is, can we go back and play it again?
I'm sorry, I apologize.
No, it's you never know how much time you need. So we always end up freezing and going back and stuff like that. Okay. But so here you can see those glass bits, by the way, are sand. So John, I should call you John?
Yeah, that's fine.
Okay. What would you prefer?
JJ, Jonathan, it's JJ's
okay.
fine.
All right, JJ, this soil, do you find it quite sandy?
Yeah, so on our farm, we have quite a different soil types around it. So this one is a sandier loam.
All right, so that's what you're seeing there. You can also see, can you see my cursor? No. You can't, can you? We can see Whitney's cursor. That's all right. So Whitney, do you want to go around the areas that are aggregates?
Absolutely.
So you see these coppery colored things. That is, bacteria is sticky. So if you don't disturb it, it will start to form what we see here, which are these aggregates. And that gives you soil structure. So it's really good to see that. And you can see they're starting throughout. Anytime you see clumps, I'm not seeing a lot of organic matter here, but I am seeing soil aggregates. So that is a nice population of bacteria that is colonized because it hasn't been disturbed.
And maybe we can also just clarify for everyone what we mean by sand. So these bits here.
Right, and you see that big there, that piece for sure.
Mm-hmm. And these sandy,
the smaller
these sandy
pieces
loams
might
are
be silt, but that is definitely sand there.
the sandy loams are very typical of Long Island.
There you go. So JJ, how long has it been since you tilled this?
Probably less than a week. And then we had a heavy rain. We're actually we just planted half of it yesterday and planting the other half for overwintering carrots, beets, onions, and things like that.
What's surprising me though is that you still have really good soil structure here in the form of those aggregates, and it's very populated with the bacteria. You're not seeing fungal because that every time you till this field, it will the fungal will go away and it will become bacterial dominant. And as we were talking about earlier, that is nature's way of selecting for weeds so that the soil gets covered. Because as Farmer Craig always points out, and I love to do his little thing, Mother Nature wants to be covered, just so we remember. And that is the key. And I talk a little bit about that later. So this is now the no-till.
Yeah, so now we've continued on to that third or the middle sample that was a bit of a darker soil. And so this is at the same magnification, but as you can see, it's already showing much more signs of life.
So do you want to point out that testate amoeba on the left?
This?
Yep. And then it looks like you've got a flagellate bumbling around there in the center. And another one above on the top there. So flagellates are the smallest predator, and it's a type of amoeba. And so it will be consuming the bacteria. So you have that nutrient cycling going on. You can see the size of that aggregate on the top left there. What I'm finding interesting though is the space in between. So the tilled field was a lot fuller. Now, what have you got growing here, JJ?
So there was, I believe it was a arugula, and we are using LI compost, OMRI listed, when we mix it in. So it's really loose stuff. Right.
So that's why we're seeing all this space. It's not condensed at all. And I have an example, which we'll look at later, where it's compacted soil, and you can see what that does. So there's lots of room here. There's lots of room for life to move around. The bacteria numbers are probably less than we would want, but it might be, did you just finish growing food? Is it done?
Yeah, it's pretty much on its last leg, ready to be cycled out.
Okay, because bacteria and really want to be around the roots of the plant, and I'll explain why later. But it's nice to see all of this life. And again, they're busy consuming the bacteria, which is another reason there might be less in the sample. That one guy that's spinning around, I have a sample of him. That looks more like a ciliate or a forticella, which I will show you later. So this is some more of the flagellate activity.
So now we're beginning our transition into the sample that we pulled from the hedgerow. So you're seeing a different consistency in the soil structure again, a higher sand content perhaps, but still quite a bit of life.
So what's interesting, the more I'm doing microscopy, the more what happens above ground is reflected below ground. So you see how different this is. There's no consistency, where when you saw a tilled field, it's very consistent throughout. So you'll have larger pieces, you'll have organic matter, you'll have a little bit of life here and then some way over there. So and look at that, how this is teeming with life. And there's plenty growing here, right, JJ?
Yeah, we have clover mix in that hedgerow specifically right now.
Right. So we're zoomed in really tight here, but you can see it is very full, and you have those aggregates being chomped on by the flagellates, and it looks pretty sandy. There are some sand particles there. But the inconsistency, I think if you zoomed out again, you would see how there's no uniformity, because it is a hedgerow rather than a tilled field. I like Whitney's comment, it's alive. Where was that piece of fungal hyphy found?
This still is from the hedgerow as well.
Okay.
This,
So we'll talk a lot about fungal in this next talk. So I'm gonna, next samples that I have. JJ, do you want to talk a little bit about what your plans are for the field?
Yeah, sure. So our plan is to run this trial and to eventually convert the farm over to mostly or entirely no-till organic farm. So on the one field, it's a controlled, conventional, heavily tilled. One thing we do not do here anymore is plow. They've relied on the plow for probably the last 100 years for all their tillage methods. We do a very low till, minimum till with a vertical tillage disc system here. And then we also utilize our spader as well, just for conservation tillage methods. And then on another trial, we'll be doing a solarization. So we'll be covering the entire field in a black fabric, solarizing it, just trying to let that organic matter sink right back into the ground. And then another one, we'll be doing a roller crimper, semi-permanent bed system, and we still have to purchase a roller crimper, but the idea is to really see the struggles, the successes, and then figure out for what our farm specifically is going to work. And I think that's the main thing in no-till systems. There's not really a right way or wrong way. It's that there's got to be something specific to your farm. Anything else?
I wonder if anyone has questions about what you're doing there.
Sure.
Anybody? Hi, Brenda.
Hi. Sorry, I don't have enough bandwidth right now for my camera.
That's all right. Would love your question.
I just got here.
Hi. Oh, did you have a question?
No, not yet.
Okay, you'll get back to us. I'm curious, when you're talking about your tilling method and the machine you're using, JJ, is that why it almost looks like no-till when I see that microscopy? That you've maintained the structure?
So probably on the tillage systems that we're using, a very shallow tillage system. We're not trying to disturb more than 8 inches at the most. Other than that, that's to me is quite a bit of a disturbance if you go farther than that. And since again, the plow has been the main thing here, there is a plow pen that is established. So we're really trying to rebuild those top layers of topsoil here that are really not here. Right now, the farm is in kind of a rebuilding phase. So utilizing regenerative practices to actually rebuild the topsoil that we need to keep producing and keep going another 100 years is going to be key.
JJ, one question that I get from farmers on Long Island who are still maybe in the learning or adoption phases of no-till are how do you treat the incorporation of amendments in a no or low till system?
Sure. So in a low till system, you can use things like a vertical tillage disc or some type of soil conditioner system that has a minimal disturbance on it. So sundown soil conditioner with a roller basket can do wonders while working that in. And also what we'll be doing here is a fertigation system. So direct injection through drip systems is going to be probably the easiest way for us to incorporate a lot of our fertilizers. being that liquid fertilizer usually is a little more expensive, I would say on a no-till or minimum till system, try to work it in even while you're weeding, just disturbing that ground a little bit.
And another question, or another thing that comes up in discussion, at least with my Long Island farmers, is this observance that there can sometimes be like a latency period when you transition a heavily tilled farm to a no-till system.
Like, what will the struggles be?
Well, so in particular, we were speaking to Debbie Aller at our last farm field day around soil health last week. And she mentioned that sometimes when you do a soil analysis, the numbers on a more recently tilled field will look higher as you transition to the no-till system. And some of the no-till fields in their first year or two of no-till treatment might have lower performance on a soil test, but eventually there's an adoption, or there is an acclimation, let's say, of the soil where ultimately you see a greater benefit in the long run from a no-till operation.
So I would say the transition from going, giving the soil, all this air, all this movement, all this incorporation is going to be kind of a shock, as if you're taking away a lot of those things immediately. And you're going to see your no-till probably within the first maybe 3 years really not performing the way you want it to. And that's going to be also a decision that a farmer needs to make as an investment. Will I see the benefits of no-till? Is this going to work for my system and my farm? And how will it work in the long run as well? I really think the farmers need to really sit down and figure out their goals on their farm specifically and see, is no-till the answer, or is it a temporary fix?
And then I've also heard that sometimes doing that transition incrementally can be the best pathway for a farm rather than transitioning every field all at once.
Yeah, I would say transitioning it little by little would be the best way. As you get better at it and you see the struggles, the benefits, and things that work for each field individually as well. Like here we have 5 different soil types. So each field's going to need different things for it specifically. One's going to like a roll down, one's going to like solarization, and one's going to like, a permanent bed system with cover crops on the edge rows and all that. So seeing what each field likes is going to be better. If you go to an entire no-till system out of the blue, you're going to shock not only the ground, but also probably your bottom dollar.
Monique, what do you think about showing us a few more soils?
I'd be happy to. Thanks, JJ. I'm really so thrilled with the work you're doing there, and sharing your practices is really what it's all about. And Whitney, I'm so glad that you brought us together to do this. So I just wanted to mention that I'm really thrilled, and I just learned a lot from you, JJ. So thank you.
No problem. Thank you.
All right, so we're going to geek out a little bit on some microscopy, but first I just want to put it in context. So I'm going to share this. I just have a few slides here that I tend to show, and you let me know if you see this slide with this really yummy lettuce. Get us all ready for dinner. Yeah, okay. So as Whitney putting this program together, Regenerative Agriculture, a view into regenerative organic soils. And so a view into like zoomed in like 100 or 400 times, which is what we're going to do. Now, this is a true hero of mine, Vandana Shiva, and she travels around the world and has been here several times, Ridgefield, Connecticut. And her wonderful quote, "In nature's economy, the currency is not money, it is life." And so the life, in this case, that you cannot see below ground. Some scary statistics, that one of the main reasons to do this is to grow nutritious food. And so what we're finding is with these conventional methods, that the mineral and vitamin content has dropped so much over the last 50 years. It's pretty scary. You can see in terms of specific nutrients density and the percentage of decline. And Organic versus conventional is quite shocking as well. So the more chemicals you put on there, the less that plant has the ability to take up whatever, anything except what maybe you give them, which might be NPK in a lot of conventional farms. So when we're talking about soil health, we're really talking about the continued capacity of the soil to function as, and I love to say this, a vital living ecosystem. And when I studied with Dr. Ingham, she stressed that this is a soil food web, like we have the animal kingdom above ground, below ground. There's this whole soil food web that we're just really starting to learn about. And that's really what sustains us. So again, very simple, the NRDC talking about dead and living soil, bare soil, erosion. Those are things that happen, the increased chemicals that lead to and are the result of dead soil, versus living soil, where you're talking about earthworms, you have residue, which is what JJ was talking about of the previous crops, which is really helpful when you're doing things like roller crimping, and you keep the previous residue there. And the microorganisms that we're going to take a good look at. Cover crops, of course, which JJ talked about, improving the soil health and adding nutrients. I think this is where I wave my arms around a little bit. Bionutrient Food Association, if you want to learn more about life in the soil and the minerals and the whole complex world down there in terms of soil health and food health, bionutrient.org is a wonderful place to go. And so what they talk about, and what Farmer Craig, who I talked about earlier, he's actually in Mystic. He runs Coogan's Giving Farm, Giving Garden. Everything he grows, he donates. He's a 13th generation farmer, brilliant. Anyway, he also talked about this, where in nature, plants produce a sugar, they do this photosynthesis thing, and then they feed 50 to 70% of what they take in, they send out through the roots to feed the microbes. And so the minute you start putting down chemicals, the plant goes, okay, just feed me intravenously. It doesn't bother to do that. The microbes go away, and then the plant is dependent on the chemicals. And it's also weaker. So then guess what? You need to add the pesticides, the herbicides, all of the other things, because the plant can't take care of itself. So basically, these sugars, the microbes use that to reproduce and get the minerals and different nutrients out of the soil and feed those back to the plant. And that's what we're talking about, the symbiotic relationship between the plant and the life in the soil. And that's what we talk about when we talk about soil fertility. So this is available, this book is available online, Adding Biology by Dr. Ingham. And she obviously puts the cakes and cookies right there at the roots, because that's how she describes it. And because they are sugars. And she talks about all of the benefits of that, protecting the root system from diseases, enhancing the nutrient cycling, and really creating a stronger plant with that symbiotic relationship. This is another mentor and amazing, knowledgeable professor, Zach Bush, and he talks about rototilling as being as equally damaging to the soil, spraying chemicals. He also says one application of chemical fertilizer can kill half the life in the soil. So that's a staggering statistic. Again, ways we can, all these things that we're learning about regenerative agriculture, get away from the monoculture, growing all diversity of plants, keeping as much of the soil with living roots as possible, either with perennials, with ground covers, then adding cover crops, intercropping. So if you see soil that's dead and lying there and with nothing growing, not what you want to see, at least put down a mulch, cover it up. So don't let it just sit idle. And here's a perfect example. It's losing carbon, it's losing vitality, and there's nothing to keep the life in the soil, because there's no symbiotic relationship that can be formed. So cover crops, excellent way to feed the soil. You know, when you're growing crops, you're taking out this, you're growing to add nutrients back. Here's a quick example, a sample of what the soil food web looks like. And we're going to be looking at various things. I know we're the bacteria, fungi, of course. We saw some flagellates, amoeba in the sample. We're going to be seeing all of these other guys as well, these soil microbes. And we're also going to talk about fungal to bacterial ratios. So generally, whenever you're tilling or bare soil, it will be 100% bacterial. And as I mentioned, that's nature's way to have something growing as quickly as possible, and weeds are the fastest thing to grow and to cover the soil. So that's nature's way to try to fix itself or correct the problem. If you go into an old growth forest, you're going to see pretty much all fungal. So as you move towards the tree line, you'll get more fungal in your soils. What we want to grow, though, is a balance of fungal to bacterial, which is very hard to find. Here's an example. I don't know why it did its own thing, moving to the next slide. Anyway, you can see down here, this is more bacterial, which are brassicas and things can handle. And as you move towards the woodland edge, you'll want, like strawberries and other shrubs perennials will want more of a fungal soil. So a very quick example of a trial that I did last year, I guess. I just took to a pot, and just as a very clear example, where I took soil that had chemical fertilizer in it, then I took organic soil and added some worm castings. Same number of seeds applied. This is May 17th. By June 2nd, I'm harvesting lettuce. Look at the color difference. They're exactly the same side by side throughout. You can see the harvest difference, and chewy, stringy, tasteless lettuce versus crunchy, sweet, delicious lettuce. And then here we are a few weeks later, and pretty much nothing in maybe the marigold surviving, but pretty much everything else is really not doing well. What does that look like under the microscope?
400 times.
Oh, it's going to tell us.
100 times magnification.
Sorry, I didn't know there was audio on here. Let me go see if I can go back. Here we are. Okay. Okay. There we go.
We do have a question, Monique, if you'd like to
Oh, sure.
take a question before we start the video.
Yeah.
So the question is, can annual vegetables such as brassicas do okay in fungally dominated soil?
They would prefer it more bacterial dominant, but sure. But the thing is, if you have fungally dominated soil, that's great. That's what we're all aiming for. Most of the compost and soils that I'm looking at are pretty much bacterial dominant. It's not until you really get into the woodland edge, which is not necessarily where you're going to be growing your brassicas or annuals. So every plant, if you will, has its place. So if you have an open area with the soil that has been disturbed, the brassicas are going to do better there than on a woodland edge where it might be shaded and not as bacterial dominant. Does that answer that question?
I believe so. Thank you.
I'm getting the nod from JJ, so I'm going with that. Okay, so here you can see there isn't a lot going on. This is that the chemical soil. What I find interesting, there's something spilling around there. I don't know what that is. But what I find is there's no aggregates being formed. So this is from that sample, and it's pretty barren, if you will. And this is the other side.
Organic soil.
All right, just gonna. There we go.
This is a nematode, bacterial feeding nematode.
All right, so you can see the different soil structure there. Same plants and everything, but it makes sense, right? When you're looking below ground, that this would be the one that has a thriving plants. It does have some fungal in there, great aggregate structure. So it really is the soil that determines the health of your plant. Okay, so that is the end of that slideshow. I'll stop sharing there. Were there any further questions about what we just looked at?
No further questions at this time, but once we go over the resources that you've provided to us, I think it might be interesting to revisit the video of JJ's soils and point out some of the things that we noticed.
Cool, yeah. So what I'm going to do now is share screen again. I have a bunch of soil samples that I put together that will give you an idea of, let me just go to the correct one here, of what I've found when farmers and gardeners have sent in samples. So these are our random, this is from a farm. Can you tell already what type of soil this might be? Anyone want to hazard a guess before I give it away? All right, we're just going to let it roll. Undisturbed. So this is undisturbed soil right beside a field. And so compare that to this tilled field, same magnification. So as you would imagine, it's pulverized, if you will, it's broken down. And this is years and years of tilling. And when you zoom in, there's plenty of bacteria. There's some soil structure, smaller aggregates, no fungal to speak of, although we did find quite a bit in the hedgerow. And then you have the ciliate. It was quite wet. So you see that ciliate running around in the background? Those ciliates like it when it is a wetter anaerobic condition. So, so that wasn't great to see. Now this is a little bit different. This is an organic field that was tilled. And again, this is soil that where chemicals have been applied. So totally different feel with no life whatsoever. Here is a picture of soil that's been compacted, and you can see how closed in everything is. And JJ and I talked about that a little bit, where you have so much bacteria, but there isn't really a lot of room for anything else to, this is 400 times magnification. It's just all chocolate block. And it also, there's no, I'm just gonna stop it there. Go back a little bit. There's no opportunity for the aggregates because they've been pretty much beat up and broken up. So a little bit about fungi. That is a fungal hyphae. And generally when we're looking at fungi, what we find is the longer, the darker, the fatter the fungi, the more mature it is. So seeing this fungal hyphae in what we can see are these wonderful aggregates is really good to see. This version here, this is an untilled greenhouse where they're growing ginger. In fact, I think we talk about it a little bit later. And it has been undisturbed. Just by shaking the test tube and putting the drop down, it's broken up the fungal. But it was amazing looking soil, and the ginger is doing phenomenal in there. And they've made a point of not tilling in that greenhouse. Here's some fungal dominated soil, and I think I included it in the next section. If not, remind me to talk a bit further about what this would be useful for. The other thing, a lot of talks go on and on about compost tea. And so what we did with that fungal dominated soil is we incorporated it into a fungal compost tea. And you can see this is now the tea that we're going to apply. And where we're finding this really helpful is, for example, a farm we did with I'm going to go back to that again. A farm we started in the Berkshires at the Hickories, not the Hickories, sorry, Greenagers, where the land was given to us, and then we brought in a permaculture farmer who has planted hedgerow on rows and rows of fruit and nut trees with silvopasture underneath. So similar to what Jono does at his farm, I won't get into specifics, is he lets chickens pasture underneath. In this case, we had sheep underneath the trees, and we made a compost tea from a mature fruit and nut trees and added that and inoculated the saplings with that. with the idea that we are introducing the life and the fungi, which can establish in the soil around these trees and start feeding those trees right away and giving them what they want. So here we are at Jones Family Farm. This is an established blueberry field. Now let's take a look at that soil. So you can see it's consistent. It hadn't been disturbed. We're seeing some good fungal in there. I don't know if we're going to be showing that part. There's some, here's a piece here. If we're looking at 400 times magnification, so you have some good aggregate structure, another piece of fungal hyphy there, and these are all aggregates. So it's quite well established. We saw that it found this testate amoeba, and what else do we have in here? So these are more fungal hyphy. So it looked nice and established, and the strawberries were doing really good, really well. All right, I do want to have Cynthia talk about this. She was trained recently on microscopy. She's the executive director of Northwest Conservation District. So I'm just going to let her talk about her very first soil sample from her garden.
I'm Cynthia Rubinowicz, and we're looking at a sample of soil from a raised bed area in my garden that is growing onions. And we are looking at a super cool micro arthropod, and I'm just really happy to see him in there. And it just indicates that we have a good level of organic life and activity in the soil, along with some fungi and nematodes that we've seen, lots of bacteria. We have a lovely fungal hyphy across the top of the view, the image here, and we have another nematode, which I'm pretty certain is also bacterial feeding. We are looking at 100 magnification at what is very certainly a nice long strand of fungal hyphy with evident septa along its length, and it's very straight, and oh boy, it's so nice to see that in there.
Okay, so that was it. The only reason I kept this guy here is, Whitney, remember in the video you showed, there was something that looked similar to this. This is a vorticella or a stalk ciliate. So I'll just play that so you can sort of compare it to when we go back and look at JJ's sample. So it's on a stalk and it's sort of bumbling around, and it is a type of amoeba that you will find. I'm going to stop sharing again. Okay, so what do you think?
Thank you for that. So we did receive another question in the chat about how to prepare a soil sample for observation. And so I can describe that process a little bit. So with JJ's farm, we selected 3 samples from 3 distinct fields. I collected about a cup of soil for each soil sample and captured it in what we call, for lack of a better word, a goldfish bag, which is a breathable plastic bag, because it is important to make sure that the soil stays moist, but is also allowed to breathe. I think Monique brought this up in an earlier video, which is that if you have an anaerobic soil, the soil microbiome actually changes. And so therefore you want to make sure that you capture the most accurate representation of the soil as possible by allowing it to breathe until you can dilute it. We dilute it in a test tube. It's a 1 to 10 dilution. And then we take a drop of that and put it on the slide before viewing it under the microscope. And in the future, if you know, the world makes it a more feasible to meet in person, we can certainly look at some of these soil samples under a microscope together.
Yeah, so you're doing microscopy now. So you did the microscopy for JJ samples.
I did, yes.
Yeah.
Awesome. So maybe we'll put JJ's soil video back up now and just review so that we can maybe as a group more comfortably identify some of the things we learned about today. I will share my screen. Okay. So again, we have the tilled sample all the way to the left, the no-till sample in the middle, and then the hedgerow or the untouched soil on the right.
So to me, if you compare that with the other conventional, or not, sorry, the organic tilled field, this looks really good. I looks a lot less disturbed. So I don't know if it's the method of tilling. You still have some nice aggregate structure there, and it's sandy, which helps. You know what I would love to see is a little more organic matter.
Me too.
Yeah. So by adding these cover crops and having that residue on there and leaving it, first of all, the residue underground will be food for the microbes, and on above ground it'll cover the soil, and as it decomposes will add the organic matter.
Yep. Yep.
So Monique, I just wanted to clarify for everyone, since we were talking so much about bacteria earlier, these sort of smaller bits that seem to be filling in the empty space, for lack of a better word, those would be bacteria?
Yes. And bacteria are also, and you pointed to that aggregate before, making up that aggregate there. So as opposed to conventional soils, which for some reason the bacteria are not sticky anymore. I'm not sure what the problem is, but they just sort of shiver in place. What happens in natural organic soils is that they will form these aggregates, and the more mature a soil and less disturbed, the more those aggregates will keep their colonies, if you will.
So I just wanted to play this particular sample a little farther. Would this be considered a fungal hyphae here?
It might be. It's hard to tell. It could be what's left of a nematode that might have been in the soil. Because
that would make sense. We did release a lot of nematodes across our farm.
Right. You see how that tail has a distinct look about it? So yeah. Let's go with nematode. Nematode. You say nematode.
Yeah.
So that might be, oh, that's all right.
Feel free.
Well, I thought I saw what might have been a fungal hyphae in that last piece.
Oh, okay. Yeah, I'll just go back really quick. It seems like this could be a fungal hyphae here, perhaps.
Yeah, and then there's a clearer one. If you go, see that, stop it there?
Right there.
Yeah.
Yeah.
That might be a fungal hyphae. It might be a small piece of organic matter. If you zoomed in, you could get a better view of that.
So proportionately, significantly more bacteria to fungal hyphae in this sample.
Oh yeah. Yeah, I would call it bacterial dominant, but with nice aggregate structure and very full. So you have a really nice bacterial count in there. So it's very much alive.
And then when you say you'd like to see an increase in organic matter, how would that play out on the screen?
So you would see pieces that are just sort of rougher. When you saw that undisturbed soil, there was a lot of things like, what is all that stuff? That is organic matter. So whenever you can't really identify something that has a frayed edge, or it's sort of a different type of a shape, it doesn't have that coppery color that an aggregate might, you can assume it's organic matter.
All right, and then just going back to the no-till greenhouse. My biggest takeaway from this is that we have a lower sand content and a greater diversity of life. The testate amoeba again, and so on.
Right. And oh, so you see this guy spinning thing around in the middle there? He's spinning. That I think is a vorticella. You can sort of see a stalk on him. So that's why I was looking for an example of a vorticella. for you today. They're really cool. They can be beautiful types of amoebas there. But you can see everything is very active and alive, and I love seeing these aggregates in here. Lots of life. JJ, what does it look like how does it feel to look at your soil under the microscope?
It's really cool. I mean, aside from getting, your chemical readings back and all your organic matter readings back, it's actually nice to see that, we are doing the right things, that me and my right-hand guy, Chris, we're doing things that we learned kind of together, and it seems like it's working really well.
I would agree. I think you've nailed it. And I'm so curious, and I would like to volunteer Whitney to take soil, is that nice of me? To take soil samples from those 4 trials that you're going to be doing throughout the season and see how it changes the microbial life in that soil.
Anytime.
So you can see this is teeming with life. Undisturbed, sandy, but really nice and full, and plenty of life in here too. So the hedgerow also looks fantastic. Now you were explaining that the hedgerow isn't just undisturbed soil.
So in previous years, most of these hedgerows are just driving areas. So it gets super compacted. So to kind of fight that, we decided to cover crop them with clover, make it a little better for drainage, and also add, life into the actual area, less compaction. So that was our idea.
That might explain why it is tight, but there's life, and it's a lot of aggregate structure and things like that. All right, because I was wondering why it was so tight compared to the no-till in the greenhouse, but that makes sense if it's been compacted before. Because we saw what that compacted soil looked like on that other farm.
Yeah, we use a grain drill mostly to get through that. Instead of like tilling it up, we use it as a no-till grain drill.
Excellent.
So we have another question for the chat. I believe there's a couple questions in one. So is it worth making or sourcing fungally dominated compost for amending the soil? Or is adding normal compost and not tilling enough? I've heard good things about the Johnson-Su bioreactor for making fungal compost. Any thoughts on that method?
Yeah, I was supposed to do it this year. How about you, JJ?
I mean, I've never used the Johnson-Su bioreactor. I'm a little hesitant on actually, making my own fungal compost, because I've mostly made non-fungally dominated compost, just kind of what's in there. That's how I learned to make compost. So I would say it may be worth looking into it more, but it really depends on your farm. It's a hard question to answer. I'm gonna be honest.
So maybe more for the permaculture farm that's growing all the rows and rows of fruit and nut trees. They would definitely benefit. And for those of you who don't know what a Johnson-Su bioreactor is, JJ, do you want to talk about it, or do you want me to talk about it?
No, you can go ahead on that one.
So basically what they do is they'll get a pallet, they'll drill holes in the pallet that are the same size as these tubes, PVC tubes, and then they'll just get wire mesh, cover that with, you can just type it in as well, Johnson-Su, but they'll cover that with weed control fabric. They'll fill it with wood chips primarily that have been soaked in water. They have to have a lot of water content. You put them in there, you put in the, first of all, you put these about 5 different PVC pipes, and then you fill it with the wood chips. And 24 hours later, you can remove, this is the coolest thing, you can remove the pipes, and those shapes, the fungal has grown so quickly in that 24 hours, nothing is going to move. You can remove the pipes, and you'll have wonderful aeration throughout your pile. And then you just put a very simple well, I could do that overnight, not, irrigation system on top, and you let it sit there for 6 months, and every day for a minute, the little irrigation comes on so it stays moist. And after 6 months, you apparently have this amazing fungal dominated compost. So imagine you have these fruit trees, you just, mulch over with this amazing high fungal compost.
So a common practice that a lot of tree growers and berry growers will just put down the mulch on over, a lot of their tree roots, and that will help kind of boost the fungal life underneath without having to do a lot of that. And that's what I've stuck to most of my career, because doing that, it takes a lot of work to actually do it correctly.
Yeah. And then you really want to look at it under the microscope.
Yeah.
So,
Yeah.
thanks.
All right, well, we are approaching the end of the seminar. So I would like to encourage anyone else who has questions to please enter them in the chat. And I would also like just to make a quick plug for the Long Island Organic Transition Program offered by NOFA New York, which offers financial support, application support, on-farm education, farmer-to-farmer mentorship, like we have today, and generally just community around organic farming and organic transition on Long Island. And I'm going to put the link for the program in the chat. If anyone is interested in learning more, please feel free to reach out to me personally. And I would just like to say a huge thank you to JJ and Young's Farm for allowing us to take this intimate look into the soils and learn about the various no-till regenerative trials that they're doing on the farm. And then also to Monique for taking us on this journey as we learn about the soil microbiome and the importance of the soil microbiome to a healthy organic farm.
Thanks for bringing us together, Whitney, and doing all this.
Yeah, this was great.
Again.
And I'll put my email in if anyone wants to reach out to me personally. Any questions, I'm more than willing to always help another farmer with anything.
Thank you. All right, well, thanks everyone and have a great week.
Everyone have a good night.
Yeah, you too.